Ladies and gentlemen, thanks for joining us for our issue briefing on editing humans. Those of you that are unfamiliar with this format, let me briefly explain. These are very short, hopefully high-intensity sessions of 30 minutes where we aim to cover a topic in a way that possibly is not covered in other parts of this program. The reason we're here far away from the rest of the sessions is a very good one. It means that we can make a bit of noise, we can be a bit more energetic, we can get to the heart of the matter quite quickly without doing rounds of introductory remarks. So the style is going to be hopefully informal, engaged, and energetic. I'm going to just start with a couple of opening questions, but I really do encourage you all to leap in, disagree, agree, share your comments, ask questions of each other, and let's hopefully have as exciting debate as we can possibly have. So this session is on editing humans. It's a topic which is growing in the public perception. It's also a very rapidly and fast-changing area of scientific endeavor. It was only in 2009 where the first human gene editing trials began, and things were moving very, very slowly until 2012 when the CRISPR technique of gene editing was developed, which allowed a much faster acceleration of progress. Fast forward to 2017, and we have 20 human trials. I was interested to read in the Washington Post earlier this month that a Gallup survey of public opinion found that the number of Americans that found human cloning was on a par with polygamy, growing upwards and upwards, so from a small base, entering the public consciousness. And yet still, there seems to be a line between the public perception of gene editing and designer babies and actually what the endeavors of in the scientific field. And this is the line that we're trying to explore today in this session, the 30 short minutes we have. So I'm very, very pleased to get my speaking out of the way. You'll be hearing very little of me, hopefully, going forwards. My panelists here, I'm delighted to say, is Luhan Yang, Chief Scientific Officer of eGenesis Biosciences in the USA. Luhan, your organization has been working with CRISPR since 2013, so you're in the vanguard of this field. You really are. But perhaps let's start by just telling us how you feel that gene editing has progressed in the few short years that it's entered the public consciousness.
Sure. So hi everyone, I'm a scientist in training. When I started my research 2009 at Harvard, despite the fact that we have 30,000 genes in the human genome, only 2 genes was modified by that time, largely due to the lack of tool. So as Oliver mentioned, 2012 is a tipping point for our field that CRISPR-Cas was first demonstrated we can modify human genome and animal genome in a reprogrammable way. And our group first demonstrated in human cell it can happen in the early 2013. After that, the field exploded. So I think so far, CRISPR-Cas has been applied to more than 30 different species, including animal, plant, and human being, and probably it's used in major labs in academic setting or industry. So I want to also comment on the title of today's talk, editing humans. I think it's kind of provocative. When people talk about editing humans, we are thinking about embryogenic editing, but in the scientific community, we are truly thinking about how to utilize a tool to address some huge unmet medical need. For example, this tool has been used in clinical trial to enhance the efficacy of the immune cell for the cancer therapy to treat those cancer patients. And we are using such a tool to treat some blind diseases, some genetic diseases. And as for eGenesis I'm leading right now, we are trying to modify the animal genome using CRISPR-Cas so that we can deliver human transplantable organ to serve millions of patients waiting for organ.
Now, how close are we, just to get an idea of the pace of progress, how close are we to actually using this technique to end disease, inherited or otherwise?
So I think we are still at the start of the revolution. To put this in perspective, every drug from research to patient will take, in average, 12 years and $2 to $3 billion, even small molecule. And CRISPR-Cas is a very young tool. It's only 3 years old. So there are a lot of things we don't know. As a scientist, there are 2 things we care most. One is efficacy, and the second one is specificity. And I think we have already demonstrated it can be very efficient, but specificity is a concern because even one off-target hitting other places of the genome will be detrimental. It can be tumor, it can generate dysfunction. But for different indication of diseases, there are different balance of the cost and benefit. For example, for those cancer patients, maybe they can tolerate some of the side effects, but for normal monogenic genetic diseases, if there are some alternative medicine and specificity is not to the standard we want, so there are higher bar and probably requires additional effort to make it working and safe.
We'll come back to the discussion on the provocation or otherwise of the title. Again, I'm interested in the pace of progress to start with. And there's been some regulatory milestones passed, as well as medical milestones passed. I mean, the National Academy of Sciences in the US in 2017, in February, I believe it was, put down another milestone on the way to mainstreaming this technology. And I'd be interested to hear your views on where this trajectory is coming from. Is it a response to more rapid progress in other countries around the world, or is it more to do with a growing realization and acceptance, possibly the growing public acceptance of the technology?
Right, I think there are a lot of expectation on the technology global-wide, but I think because of the communication and because of the open resource of the scientific community, now it's a consent in the global community. We should do that as a very responsible way. But I have to admit that the scientific progress is always ahead of the public education, communication, and perception. So it's our responsibility to deliver a responsible message to the public to indicate where we are, where the future holds. I think for every genetic diseases, there would be some gene editing component can be very effective, but we are still at the very early stage to understand the long-term consequence of the tool in human body. And it probably take years and a lot of setback for us to get there, but it truly provides an alternative treatment to medicine.
Maybe it's a good time to ask around the room and see if we've got any questions or comments. Please do stick your hands up. We have microphones at the ready. Okay, it's very early in the morning, obviously. So there's clearly work to be done on the public side, and it may well be that the public debate never catches up with scientific progress. But what kind of job are you doing as a community to put the public perception, bring that public perception up to speed? How would you mark your progress as a scientific community?
Right, so that's probably one of the reasons why I'm here. I'm glad to take the opportunity to share the progress and share with you where we stand. And we also engage a lot of entertainment industry. For example, there's a book coming out next week by an American writer, Ben Merrick, about woolly mammoths. So while we were at Harvard, we're trying to using gene editing tool to revive woolly mammoths as a demonstration of the concept that we can help some distinguished species so that can really liberate people's imagination where the technology future holds. And another way is publication, because publication, if a paper can publish in some top-tier journal, it has to sustain the scrutiny of the expert in the field to make sure it's accurate in terms of the data and in terms of the claims. And that's another way we can engage the public.
And there was I hoping that the woolly mammoth would actually be brought to life for real. Slightly disappointed to hear. Gentleman there in the third row, please remind us where you're from and give us your name, please.
Yeah, John Corrigan, Wall Street Journal. Thanks for coming today. Can I ask you, so talk about gene editing and the potential, not necessarily what should be done, but what could be done. At the outer limits, what is the potential? How could humans be changed through this process of gene editing?
It's a great question. And I think we are only constrained by our imagination, what a tool can do. And if I have to characterize, I think there are three major applications of the gene editing tool. The first one is gene therapy to cure some genetic diseases. The second one is agriculture, and whether we can use such a tool to enhance the productivity and the nutrition, the safety, as well as the production of some livestock and agriculture plant. And the third one is to engineer some feature of the organism, offer some new functionality, such as xenotransplantation we are working on, to use pig as a bioreactor to mature organ for human transplant.
What is the most exciting area for you of all those?
For me, I'll be very proud if we can make tangible progress for xenotransplantation. So I think for genetic diseases, due to the cost of the R&D, maybe once it becomes a medicine, it can only be accessed by a few rich patients. We have to be brutal. Whereas for organ transplantation, people always know there's a shortage of the need, but the magnitude of the shortage is kind of striking. So in the US, about 100 patients waiting for organ, only 1 patient can get transplantation each year. And the situation is worse in Asia. Like in China, there are over 3 million patients need organ, but there are only 3,000 donations each year. So human life is at stake, and we don't have any medicine solution. And the concept of xenotransplantation is to harness what nature has created for us, the organ, and tweak in a way using gene editing tool to make it human compatible. And that is the thing make us very excited every day to work, and also the sense of pride, even we know it's a long haul.
So in effect, xenotransplantation is democratizing the outcomes of healthcare. You're bringing healthcare and making it accessible to more and more people in this way.
Yeah, exactly. To provide solution to the people who otherwise couldn't get the resource for organ transplantation.
I want to go into the limitations soon, because you do hear of failures and setbacks along the way. But first of all, let's go to that gentleman in the back row, please. Just wait for the microphone, sir. It's a bit of funny acoustics in here.
Hi, I'm Ben Harburg from MSA Capital. I was interested, historically in China, particularly in the genomic space and gene editing, there's been some arguments that it's more of kind of brawn over brain, so a lot of volume and a lot of work being done, huge orders of sequencing machines, but not a lot of kind of cutting edge technologies and processes being developed. I was wondering, particularly in gene editing, and we see a lot coming out of the Seattle area and other innovation hubs in the US in gene editing. What kind of new cutting edge technologies or changes are you seeing where China is starting to lead the way in innovation here?
Right, exactly. Thank you for the question. I think people got very excited about CRISPR because it truly moved us from the stage we couldn't do anything to we could do something. But I think CRISPR-Cas is similar to Sanger sequencing when it was invented in 1974. We could do something, but it's just a start. And there are so many limitations of the technology. For example, specificity is not good enough for some of the gene therapy. And knocking out is quite efficient using the tool, but how to knock in or replace a functional gene with precision still require improvement. In addition, if we talk about we want to engineer a pathway or new feature, we are talking about different genes, megas of base pair. Right now, we can handle thousands of base pair. So scalability, specificity, complexity require innovation. And I imagine there would be a lot of breakthrough and new technology coming in the next 1 decade.
And let's just also develop that international angle there as well about the different countries. And the gentleman mentioned China. There are obviously different rates of progress in different parts of the world. Where are we to? Has that been overblown in the public perception, the fact that some countries are racing ahead in terms of this development, possibly due to less regulatory oversight? And I suppose more importantly, are we getting any closer to kind of an international consensus on how to move forward with this science?
I think I had the privilege to have education both in US and China, and which enabled me to do what I do right now. And I do feel Europe, China, and US are head-to-head in terms of the competition. And I do feel the revolution of genomics and gene editing do provide China the opportunity to catch up. in the biotech industry.
Gentleman there, second row, please, sir.
My name is Conrad. I work with Bioinnovators Europe, and I want to build on your question. As you may recall, there's a biosafety protocol that was adopted under the Convention on Biological Diversity in the early 2000s for the use of biotech and the environment. And my question is, what chances do you see for a similar process applied to this technology that you're talking about? Or do you think that it would take too long and technology is so fast that the UN regulatory process will never be able to catch up and contain this kind of question?
It's a good question. How can we make sure there are some global consent how to use the technology in a safe and responsible way? I think probably such an effort should be bottom up rather than top down to make it effective. And as a scientist, what we are thinking about, how can we redo what gene editing has been done or prevent some of the garage scientists perform gene editing in a non-responsible way? And there are some thinking around it. First, whether we can have the inhibitor of CRISPR, whether there are some way we can recover the gene editing has been made. And those are quite active topic in the scientific community, try to think about the mechanism to protect our society be harmed by such a technology.
Are you satisfied that there is the willpower, talking of bottom-up regulation, and does that willpower exist? Does that intent and that ambition level exist across the scientific community around the world to get together and organize themselves in such a way that such an environment could be created?
I think the competitive dynamic within different academic group or within among different countries may push us forward in a very aggressive way. But we need to remember that medicine should be no harm to the human being. That's the bottom line. And I have to say that the tool requires some improvement so that be more specific and be less immunogenicity to be effectively delivered to the human at this stage.
We're just a brief warning. We have only 10 minutes left of this short session, so get your questions and comments in, maybe from this side of the room, perhaps. Just saying. While we wait for you to formulate your ideas, let's talk about the drawbacks, because I've been reading of recent setbacks in HIV research of CRISPR and gene editing not fulfilling expectations or anticipated results. What are the limitations of the science so far, and what are the greatest challenges that need to be overcome if this unlimited potential that you talk about is going to be achieved?
Right. I think it's a synergy between reading the genome, which is sequencing, to modifying the genome, which is gene editing. Because sometimes I think the constraint is we don't know what to modify. And we don't know after modification whether there are some unexpected consequence. But the way we know is we can implement the tool in some diseases model animal, then to understand the phenotype, then we know. So it's like gene editing can help us understand more about the functionality, and we need to use gene sequencing, complement this technology for us to understand more about the biology to go forward.
And alongside CRISPR, what are the next? Are there any kind of big milestones that you expect? What are the kind of give me your view of the next 5 years going out? What are the kind of milestones that we can expect to see?
On the technology development part, I can imagine there are better tools than CRISPR can come to our society, which can have higher specificity capacity. And on the application part, right now we already started the clinical trial using gene editing to enhance the immuno-oncology effect. And potentially there are more clinical trials going on, potentially some drug being approved using gene editing tool for some monogenic diseases such as hemophilia, so on and so forth.
So a quick show of hands. Who's comfortable with gene editing? Who finds it exciting? Who finds it and who finds it worrying? So that's one, and that's 3 versus 1. That's a pretty small sample. but that's a 75% favorable rate. I believe there's a lady there in the third row asking for the microphone.
I'm a layman, so maybe stupid question. So, two quick questions, if I may. First is, in your company, when you do genome sequencing and genome editing, it has to be for certain hypothesis, right? So is it like a commercial thing? Somebody comes and actually asks you to do something, and then you do it for them. So is there any moral hazard elements there? So that's the first question. Second question is, I recently saw an article that says in the medical world, the doctors or the researchers treat diseases in a very silo fashion, like heart disease or diabetes or some chronic diseases. But the underlying theme of that article says actually it's all a result of people living longer as a result of aging. We should treat aging as a disease. So is there anything that you can tell us, from a gene editing point of view, that is there something that we can actually attack or approach this disease of aging? So two questions. Thank you.
Okay, so the moral hazard of the commercial necessity and the desire to improve human health is the first one. And then aging. What are you going to do about making us live forever?
It's easy to answer the first one. So I don't think we're at this stage we can customize gene editing, and I don't think it's responsible action to do the customization at this stage. As I mentioned, with the progression of the technology, probably we can first address some of the urgent medical need to start. Once we know about the safety and efficacy, then we can address other common diseases. And for the second one, aging, it's a very interesting topic in the biology community. And my personal view is aging is a systematic symptom or phenomenon of life. And at Church Lab at Harvard, the lab I got trained, so we are testing 40 different gene editing ways, try to see whether we can elongate the life of small animal. But I think it's a huge undertaking. And systematic approach such as anti-apoptosis, which is used by a company called Utility at Seattle, or other approach, I think would be more thoughtful at this stage.
Okay. Any more questions? Okay, well, look, we've covered an awful lot of ground. I just want to conclude, perhaps, then, by returning to the beginning of this conversation and addressing the public advocacy side of this, and what is the logical conclusion of this? Are we ever going to be able to draw a line in the sand between the work that you're doing and the understanding of this technology? What's it going to take? Is it possible?
I think we are at the tipping point of the technology revolution, and inevitably it will trigger a lot of revolution in the industry and impact our daily life. However, we are still at the early stage of research, and we welcome the engagement from the public to let us know what's the urgent need in the society and how can we do collectively to make a huge and positive impact.
Well, a fascinating discussion, which I've enjoyed greatly, and I hope you have too. Clearly, huge amount of progress has already been made, and clearly much more needs to be done. So I wish you the best in your endeavors, and I thank you for joining me today, Luhan. And thank you for joining us here this morning, and thank you for joining us live online. This session is now over.